Hello, everyone. Can you hear me well? How's the connection today?
Good.
Good.
Good, thank you.
Thank you. So it's Wednesday, 11 o'clock, October 28th. We have one more lecture to discuss the nucleic acids. And then we have a test next Monday. So we have quite a bit of material to cover. So I will get to that right away. So I already made the exam. It's already actually posted, but you cannot see it yet. You will be able to see it on Monday. And I suggest that you guys, last time a couple of you had some issues, technical issues. Make sure the equipment works. You get up a little earlier. Test your equipment, make sure it works. I will also release those practice problems for you again from last time for you, just to make sure that everything works and you can see the figures, because a couple of you could not see the figures last time for some reason. And of course, you should use Google Chrome. Google Chrome is the recommended browser for Canvas. So if you use that, you should not have any technical issues. All right, let's get to material. Let's get to the material. So we talked about double-stranded DNA last time. Double-stranded DNA, the discovery of double-stranded DNA, two grooves, major groove, minor groove. And right now, up to this point, we believe that the DNA primarily exists in these three forms, double-stranded forms. So it's not just one double-stranded form. There are a number. And so the three ones are shown here. So the most common one that is considered to be the classical Watson-Crick form is referred to as B form, where all the bases are stacked and has the dimensions that we just talked about. And it turns out that if you dehydrate DNA, you end up with what's known as A form. So in the environment where there's lack of water, right? Or for example, if you crystallize DNA, you primarily get the A form. So here the base pairs are somewhat tilted. Goes this way, goes that way, goes that way, right? And the major groove becomes slightly wider, right? And the minor groove slightly narrower. Slightly narrower. Okay. Now there's a third form known as Z form. So this one, it has some kind of a zigzag. If you look at the phosphate backbone, it has a zigzag appearance. So Z form primarily comes from when you have purine bases alternating with pyrimidine. For example, often when you have, let's say, G, C, C, G, G, C, C, G. So when a sequence is like this, so let's say this is 5' to 3', right? And this is 3' to 5'. When the sequence is like this, so this is conducive to formation of the Z form. And so here are some of the characteristics of each of these. So both A and B form are right-handed helices, and Z form is left-handed. Left-handed. Now diameters are different, and base pairs per helical turn are different. So B form, so last time we talked about, it's about 10.5 nucleotides per helical turn. So A form accommodates a bit more of these nucleotides per helical turn. And so it's possible that the reason why you can kind of, it's kind of, DNA is kind of squashed a little bit, right? So basically you take a piece of DNA and you squash the bases along the double helical axis, right? So it's possible because this is dehydrated form, maybe that's what's happening. So water escapes from the double helix and the bases find each other closer, the bases become closer to each other. So that's a possibility. Now there is different sugar-Pucker conformation. And the main difference is actually in this glycosyl bond conformation. So in A and B form, it's anti, right? So remember we talked about that. So so this, let's say when it's this way, it's syn. When it's this way, it's anti. So it turns out that for both A and B form, it's anti, but for the Z form, so purines are still, sorry, so for pyrimidines, it's still anti, but for purines, it will switch to syn. So the purines will find themselves above the ribose ring. So as far as the physiological significance of each of these forms, that's not clear. In fact, A form is still it's still highly debatable whether the A form actually exists under physiological conditions, or perhaps it's just an artifact. When you isolate DNA from the cell and you start working with it, it undergoes some dehydration, and then you get the A form. Perhaps in the cell, when it's fully hydrated, the A form never even occurs. So we don't know that, right? Because we don't have the, unfortunately, we don't have ways to visualize the DNA conformation in the nucleus in the cell at this point. All right. So these are primary forms of DNA. And so as far as the sequence, so obviously the two chains will differ in sequence, they're complementary, right? They're complementary. So if this is CAAAT, this will be GTTTEA, and the two chains run anti-parallel. So remember the direction, the sequence is read from 5' to 3', and this one will be read from 5' to 3' this way, right? And this kind of anti-parallel arrangement immediately suggests how DNA is replicated. So if we go to the next slide, so you can see here, obviously when the DNA is double-stranded, you need to separate the two strands from each other, right? To separate the two strands from each other, so the genetic information can be read. And so you can see here, so if this is the parent, the original strand, then the new strand will basically start at the point of the separation and start to be synthesized from the 5' to the 3' end, right? And so you can see this one will be synthesized from the 5', which will be on this side, going up. So this is the direction of the synthesis for the daughter strand. And using the parent strand on this side. And this will be the direction of the daughter strand synthesis for the parent strand on this side, 5' to 3'. Okay, and so you can see immediately already, so it's actually beautiful how this works, right? So you don't really, it's not like you have to separate the two strands fully apart and then synthesize two more, then re-anneal them or recouple them. You don't have to do any of that, right? So basically immediately you have one parent strand starting to separate and two daughter strands are starting to be synthesized, and they synthesized immediately adopting this double helical conformation, right? Double helix parents and two double helices daughter strands. So very conservative, very straightforward process for DNA replication, right? So nature loves simplicity. The simpler it is, the less opportunity there to make a mistake. And with DNA, you have to be very careful, obviously. Mistakes can be costly. All right, so DNA is synthesized using the enzymes known as DNA polymerases. Now this, obviously you need nucleotides, which will come in and insert themselves every time for each step of the ladder. Now also, now there are the enzymes which make RNA. When you need to make proteins, the process is similar. Only instead of making DNA, you will make the, sorry. You will make the messenger RNA. RNA polymerase. Okay. So DNA polymerase and RNA polymerase. So these are two main enzymes which work on DNA to make complementary strands or the strands which are made of RNA. Any questions? All right. So let's talk a bit about RNA. So, so DNA is responsible for storing the genetic information. And so RNA is primarily going to be the carrier, right? So we're going to take the genetic code and we're going to carry that to the ribosome to synthesize proteins. So that's the main function of messenger RNA. So, so synthesized using DNA template and generally occurs as a single strand. So RNAs don't form double-stranded sequences except for specific regions, which we will look at in a moment, which are called inverted repeats and various shapes, such as known as hairpins. So we will look at that in a moment. So obviously ribose instead of the deoxyribose, uracil, no thymine. And it's important to know that one messenger RNA may code for more than one protein, right? So when DNA is transcribed into a messenger RNA, that messenger RNA piece may have a number of genes. And so messenger RNA and transfer RNA basically will bring the genetic information from DNA to proteins, right? So messenger RNA will bring the codes and transfer RNA will bring the amino acids and read the codes in messenger RNA to select specific amino acid for a particular position in the synthesized protein. And so here is a space filling representation of a messenger RNA. So you can see here in yellow are the base pairs. So there's some stacking here going on. So there's some stacking. Now orange supposed to be phosphates. I think maybe I'm a bit colorblind. Is this orange? That color there?
Yes, it's orange.
So this must be the phosphorus of the phosphate. And then the green are the oxygens, I guess. Oxygens of the phosphate. So forming the phosphate backbone, going like this. So it's also a right-handed helix, but it's single-stranded. But in general, RNA adopts variety of structures. So here is some terminology which is used. For example, when you have a piece of messenger RNA that has only one gene in it, yeah, one gene, meaning that it encodes for one protein, then it's going to be monocistronic. And when there is a number of genes, so this will be polycistronic. So in this case, a number of proteins can be synthesized from one particular messenger RNA strand. All right, so one possibility for the RNA to form these variety of other structures, such as hairpins, for example, is the repeats. So repeats among the nucleotides. So for example, one of the most familiar to us, which doesn't have to do with the nucleic acids, are known as palindromes. So palindromes, for example, are words or phrases which are the same when you read backward or forward, right? So for example, civic, you read this way or this way, it's the same word. Race car, same word. Rotator. Now there's a very long one. If you read it either way, from either end, it ends up being the same word. So it's a Finnish word for soap cup batch trader. There's even an ancient Greek text shown here, which means wash the sin as well as the face. Okay. Now, so these kind of palindromic sequences can occur in RNA, for example. So you can see here, so this sequence is you read it from this direction to this direction, and this sequence read from this direction to that direction are palindromic, right? So they're symmetrical. And so it doesn't matter which way you do it, from left to right or right to left. And when you have these kind of palindromic regions, then what that means, usually then the complementary strand here can undergo double-stranded region formations with the sequence on the same strand. Okay, so now there's also mirror repeat. So these are not as important in terms of the tertiary structure, but you can see here, this is the palindromic sequences on the same strand. So how does this work? So this is a hairpin. This is a hairpin. So you can see here, This is 5' end, 3' end. And so this would be TAGCGT, and this will be the complementary sequence, right? So this will be an inverted repeat where in the 5' direction, you have sequence which is complementary to the sequence on the in the 3' direction. So they can undergo double strand formation, right? And that will form something which is known as a hairpin. And so this loop here, now this can have a number of different nucleotides, right? So these nucleotides don't necessarily have to base pair with each other. They could be anything, right? Because these don't interact with each other, just a loop. Just a loop. What's important is that you have complementary base pairs in the actual interaction between the inverted regions. So hairpins, and you can see there are lots of hairpins. So this is the structure of a RNase P, I think, if I'm not mistaken. RNase P, yeah. So basically, RNase P functions in the processing of transfer RNAs, and you can see here that there are a bunch of these double-stranded regions. So these are hairpins, right? So for example, this hairpin only has actually 4 nucleotides in the loop region. This one has many more, right? They can be long, they can be short. So lots of different hairpins. Now, one important non-canonical base pair which is formed is GU, right? So we know GC is the one that is the predominant one, which is the Watson-Crick. But uracil can also participate in the formation of base pairs with guanine. And you can actually find, can you find GU anywhere here? GU base pair?
There's one in the bottommost hairpin.
Bottom, this one? This one?
Even lower.
This one? GC. Where did you see it?
Your mouse was just over it. Let me see if I can annotate it.
No, I have to share. I have to give you. Oh, good job. Yep. Perfect. Yep. And then that's GU. Now there is a. I saw earlier. There is another one here. Right. So they do occur. They're not obviously not as this is another GU. They're obviously not as common as GC, but it does happen. So one thing I do need to mention to you guys, something that you need to keep in mind, is that even though we talk about base pairing through hydrogen bonding, and we say that G pairs with C because of the 2 double bond acceptors on one, and 2 hydrogen bond acceptors and 1 hydrogen bond donor on one, and 2 hydrogen bond donors, 1 hydrogen bond acceptor on the other, complementary. A and T or A and U, they also complementary through hydrogen bonds. It turns out that the ability to form hydrogen bonds is not the primary factor which makes the base pair formation so specific. It turns out that specific bases are accommodated by the double strand structures. For example, if you have a G on one side of the double helix, it's only C that can come in and base pair with it. So, and the hydrogen bonding is a secondary factor. right? So the sterics and the ability to basically to kind of slide into this double helix appears to be the primary factor. So there's a scientist at Stanford University. His name is Professor Eric Kuhl. He's done a lot of research in this area. He's a modern researcher. He's highly active these days. Basically, what he's done, he would, for example, remove this carbonyl here, right? So instead of uracil, he will, let's say, keep this NH in place. Keep this NH in place. But then, instead of a carbonyl group here, he would put something like, what would he put here? Let's say sulfur. So the sulfur is not very good at hydrogen bonds at all. Remember, it's only the small oxygen, small electronegative oxygen, which forms hydrogen bonds. And so, and on this side, then, so it will be hydrogen bond here, and the hydrogen bond here is going to be much, much weaker. And yet, he would show that the double helix is as stable as the one with the oxygen, or even more, even sometimes even more stable. So basically, you're removing or weakening the hydrogen bond formation, but you're not decreasing the stability of the double helix. So in other words, the bases are still paired very strongly. And so the hydrogen bonding is, even though originally when the DNA was discovered, everybody thought it was all about hydrogen bonds. And actually we still teach that. And in fact, the most notebooks, sort of most textbooks are still written that way, that Watson-Crick hydrogen bonding is a driving force in double helix formation and so on and so forth. It's important to stay updated with modern research, which shows that actually hydrogen bonding is important, but that's not the main determinant factor, right? It's just the shape of this nucleotide, which fits nicely into the double helix. And here it will be against guanine. All right. Now there are other types of hydrogen bonds that can form. So this is already common for DNA, for example. So you can see here, so this is guanine and cytosine, right? So there is 3 hydrogen bonds, just like we discussed, Watson-Crick hydrogen bonds. But then you may notice that this is not it. In the guanine, for example, there are 2 other positions. There's a lone pair here, and there's a lone pair here, right? So there are 2 hydrogen bond acceptor positions in guanine, and those are not wasted. So oftentimes you can have a third strand coming by, and if there are possibilities, or so there are possibilities which is on the third strand there'll be, if there is a base that has hydrogen bond donors, it can form hydrogen bonds with the guanine that already has hydrogen bonds to cytosine. So guanine actually participates in 2 sets of hydrogen bonds, right? One with the original strand, complementary, and one with the third strand. So DNA, that way DNA can actually form structures which are larger than simple double-stranded DNA. And these type of hydrogen bonds known as Hookstein, the scientist who discovered these, his name was Hookstein hydrogen bonds. And so this will be position 7. And this is position 6, right? Position 7 and position 6. So, so this is typical for nitrogen 7 and oxygen 6 to participate in this kind of Hooke-Steen bonding. Now, there are other kinds of hydrogen bonds. So, for example, here, adenine can bond to guanine. For example, here, guanine, you can see that you put the dimethyl substituents here. This becomes, this doesn't have a hydrogen donor capability anymore, like in guanine. And so this will not form hydrogen bonds. And so it's capable of forming hydrogen bonds with adenine. That is incapable of forming hydrogen bonds on this side, right? So no hydrogen bond here. Now, in general, so you can have trimers like this. So the double-stranded would be a dimer, triple-stranded would be a trimer, and there are also tetramers. So tetramers would be can form specific structures where you have a base stacking that looks like this. So here are the 4 bases, like so. No, not a good drawing. Like so. And then, so this will be the phosphate backbone of one strand, phosphate backbone of another strand, phosphate backbone of the third, and phosphate backbone of the fourth, right? So, so this will be a tetramer. So it has been shown that DNA can adopt these kind of tetrameric structures. Obviously, now these all run in parallel, right? So you can have them all parallel. You can have two of them being parallel and the other 2 anti-parallel. So there are different orientations, right? So for 3' and 5' directions. And so for these tetramers, they often occur in specific DNA segments known as telomeres. Telomeres. Who knows what telomeres are?
Isn't that like at the end of the DNA strand? Often it has something to do with aging, like that the telomere, is that the one, is that right?
Yeah.
Where the telomeres are shrinking, like with the age of the DNA?
As they replicate, isn't it as they replicate more and more, the telomere in like shrinks? I mean, not shrinks. Like it's less identical as the original one.
It shrinks. It loses.
Oh.
Yes, it gets short. It gets shorter and shorter. Correct. Yeah. So you're both correct. So telomeres are stretches of DNA at the end of the chromosomes, which with each progressive replication, they get shorter and shorter. And you're right, they're involved in aging, for example. So it's believed that, so in fact, actually, as they get shorter and shorter, there is a critical point at which once the telomere becomes so short, once it crosses this critical point, then the cell undergoes cell death. And and there is a enzyme known as telomerase what does this enzyme do?
It rebuilds the telomeres.
That's right. It puts the telomeres back in place, adds more nucleotides to them, rebuilds them. And so this enzyme is actually overexpressed in a growing organism, right? When in a young child, for example, which is growing actively, and all the tissues are being developed, all the differentiation, cell division. So telomerase is highly active enzyme. It's necessary. But once a person reaches a certain age, when the cell division is not as active process in the body anymore, then telomerase is no longer an active enzyme, right? It's not expressed to a large degree. But for example, cancer cells, they, if you think about it, because of the constant cell division, if you think about it, so that if there was no telomerase, then the tumors would never develop, right? So you start this, the cells will start growing, the cells will start dividing, they acquire this what's it called, they acquire the ability basically for uncontrolled cell division, right? And but at some point their telomeres should get so short, then the tumors will stop growing, and the cells which cross this point of no return will undergo apoptosis, will basically kill themselves. But unfortunately, tumors found a way to express this enzyme as well. So most tumors express telomerase, and that allows them to divide uncontrollably and indefinitely, right? And so actually inhibition of telomerase is a huge area in cancer drug design right now. So obviously, if you inhibit the enzyme telomerase, then the tumors will stop growing because the telomeres will become too short. And you see, this is all based on this tetramer, on the particular form of DNA, which is present in human genetic system. All right, so that's what I wanted to say about the other types of base pairing. All right, so let's switch to DNA denaturation. So just like the proteins, DNA strands can be pulled apart, right? You can break basically hydrogen bonds. So you will not break the covalent bonds by simply heating the DNA, well, within reason, all right? Let's say if you heat to 100 degrees, you will not break the covalent bonds. And so the genetic code will remain intact. There is no ground to be worried. But the hydrogen bonds will break, and the two strands will separate. So the DNA will denature. So this is partially denatured DNA. You continue heating that, and you can separate the two strands fully apart. So denaturation may be reversible, and this process is called annealing. So then you cool it down, and the DNA will come together again, reform the double helix. So now high temperature can also be induced by change in pH. You know, change in pH breaks the hydrogen bonds, and that's also a possibility. So elevated temperatures, reanneal, temperature lowered. And so this reversible thermal denaturation and annealing form the basis for the polymerase chain reaction. We're going to talk about that in a moment. And you can monitor that by UV spectrophotometry at 260 nanometers. So in general, when you have two base pairs, when they're stacked like this, they don't absorb light very well, right? So the two pi systems interacting with each other, okay? And so this interaction between pi systems actually prevents them from absorbing light very well. So, so stacked bases do not absorb. Do not absorb light very well. And so when they separate, when the two strands separate, then the base is unstack, and the absorbance goes up. And you can monitor that. And so you can see here. So this is percent of denaturation, which is based on the UV absorption. And this is versus temperature. So Tm is known as a melting temperature. Melting. Melting temperature. And basically it corresponds to the temperature at which you observe 50% of denaturation. Okay. So, so looking then at these two curves, so these correspond to two different DNA fragments. So which fragment is more stable towards the denaturation? The red one or the blue one? Which one is not denatured as easily as the other one?
The red one?
Red one.
Red one, correct. So you can see the red one will require higher temperatures to undergo denaturation. And you can see here, you will need what, 85 degrees to denature half of the DNA fragment. And here you will need 83 degrees Celsius to denature that fragment. And the midpoint Tm depends on the base composition. So for example, high GC increases the Tm. So if you have a DNA fragment which is rich in GC base pairs, the Tm will be high. Why do you think that is? Why is the GC rich fragments, why do they have higher Tm?
Because they have more hydrogen bonding.
Right. So the GC base pairs are glued to each other more strongly because of 3 hydrogen bonds, and AT base pairs have 2 hydrogen bonds. Correct. So this is something that if you see a problem like that on test, you should be able to predict the Tm or the difference in Tm based on how much GC composition you have in one strand versus another. Also depends on DNA length. Obviously, the longer the DNA has higher Tm, right? So basically, the longer the DNA, the more base pairs you have to destroy. And also pH and ionic strength. So for example, high salt increases Tm. So you can think of DNA as a polyionic species, right? So there's a phosphate, it's negatively charged, has counterions. So the DNA basically it's a salt. It's a salt which has multiple multiple phosphates in its structure, right? And so if there's high ionic strength, it's obviously it resists, it make double-stranded DNA stay together, because when the DNA strands separate, that will work against the ionic strength. Basically, you're gonna be increasing the ionic strength even more by increasing the salt concentration, right? Remember, in double-stranded DNA and single-stranded DNA, if the 2 strands separate, you will increase that salt concentration 2-fold, right? And high ionic strength will resist that. All right. So here is the percentage of GC of total nucleotides, and you can see this very nice straight line, which shows us that as you increase the percentage of GC in the DNA strength, the higher the melting temperature. The higher the melting temperature. So you can see, for example, from 80 degrees, you can go to 100 degrees by increasing the GC content from 30%, right, to 70-something%. Okay, so this is an electron micrograph. So here, it's obviously very high resolution. So here, so these pieces of DNA are still double-stranded. So basically, what slide is trying to show us is that AT regions melt at lower temperature than GC regions. And so these red arrows shown here, so these tell us that these parts of the DNA are rich in AT base pairs, and you can see there's a strand separation in these parts. There's a 2 strands coming apart, 2 strands coming apart, 2 strands coming apart. So it's kind of loops, right? So these loops are AT rich. Just an experimental verification of our hypothesis. Okay, I was gonna remove the slide, so let's skip the slide because we're gonna actually talk about polymerase chain reaction in much more detail. All right, so polymerase chain reaction. So what this is used for is to amplify DNA in a test tube, right? So oftentimes you isolate very small amounts of DNA, not sufficient for sequencing, right? So DNA sequencing is very important for our daily life, right? Not just research anymore, for our daily life. Even, for example, to understand anything about extinct organisms, right? So for example, if you isolate some DNA from a fossil and you want to understand where this organism came from, or its origin, or its relationship to another organism, so you want to compare the DNA sequences, right? And for example, you isolate only one molecule of DNA, just one molecule, and you can amplify that molecule millionfold or even billionfold, right? Even billionfold. And then once you have that, then you can sequence it. And you can sequence and specifically know the sequence of nucleotides. And important, for example, also in forensic areas, forensic medicine, where, for example, where at the crime scene, let's say the criminal left some of his or her DNA, right? And very, very small amount, not sufficient for sequencing and comparing to the database. And so the DNA will be collected from the crime scene, and polymerase chain reaction will be applied to multiply the copies of the collected DNA. So we're gonna mix together target DNA that we isolated. Then there's primers. We'll look at those in a moment. They should be complementary to the target. We'll look at nucleotides. So these are deoxyadenosine triphosphate, cytidine triphosphate, guanosine, and thymidine triphosphate. And we'll work with the thermostable DNA polymerase. I'll tell you why it has to be thermostable in a moment. And the apparatus which is utilized is known as a thermocycler. In the past, it was done manually in a laboratory by a person. So nowadays there is a machine that can accomplish that within hours, can multiply DNA and provide you just enough copies for what you need it for. So the process actually is very, very simple. So here is the, let's say, double-stranded DNA that you isolated. And this is the region of the target DNA that needs to be amplified, okay? So you will heat to separate the strands. So you will melt the DNA. You will melt the DNA, and you will add synthetic oligonucleotide primers. So the DNA synthesis does not occur from scratch. You need primers so that the new nucleotides can be added in the 5' to 3' direction. So basically you heat the DNA, you add the primers. So the primers will not attach unless you cool it, and you will not cool it all the way to to room temperature, but you will cool it to, let's say, 60 degrees. So for example, you will heat to separate the strands to 100 degrees, and then you will cool it down a little bit, let's say to 60 degrees. So the primers will anneal. And then you add a thermostable Taq DNA polymerase. So Taq is...
I have a question. Excuse me. Are you heating to denature the DNA essentially?
Yeah, so the first heating is to denature the DNA. That's right.
Okay, perfect. Thank you.
Yeah, denature to separate the strands. That's right. To separate the strands. Right. Then you add thermostable Taq DNA. So it's an organism that lives near thermal vents, and which basically lives in at temperatures around 100 degrees Celsius. And so obviously it utilizes enzymes which do not undergo denaturation at 100 degrees Celsius, right? So enzymes which are thermostable. So you cannot use human DNA polymerase for this task, because the human DNA polymerase will not work. Its three-dimensional structure will fall apart, right? So you need to use a thermostable DNA polymerase. And so the DNA polymerase will utilize the primers and start adding these nucleotides in the 5' to 3' direction, right? This way and that way. So until this region is synthesized, right? It will still continue building the DNA further because there's no stop sequence here. It will build it all the way to the end of the fragment. But what we're interested in is actually this specific fragment, which is in dark blue. And the same thing here. Then we repeat steps 1 and 2. Again, we heat, melt the DNA, separate it, add the primers 1 and 2, add the DNA polymerase, and again, we will polymerize the DNA and amplify these dark blue areas. And you can see here, some of them extend beyond, some of them actually stop here. And eventually, as you continue doing that, eventually there will be more and more of those dark blue regions which stop on either side. And as you continue repeating steps 1 through 3, and so for example, after 20 cycles, the target sequence has been amplified about 10 to the 6th fold, right? So basically, each time it's going to be doubled. Fold. So 2 to the 20th is about 10 to the 6th fold, so millionfold. Millionfold. All right, so this is how polymerase chain reaction works. And we will We will not have time to talk about DNA sequencing, but once you obtain the DNA, then you can sequence it. And actually, I'll assign some reading for you to do for DNA sequencing. It's still in this chapter, but we will talk more about sequencing when we start talking about chapter 9. But since you already have some problems on some basic sequencing in sapling, I will give you some pages to read from the text.
I was wondering about the primers. How are the primers made? How do they get them? Are they just like markers or something, or do they?
Yeah, primers can be chemically synthesized. It's primers are not very long. Maybe, to be honest with you, I forgot. Think about like 9 to 10 nucleotides. I'll double check, but think at this point I'll give you the number 9 to 10 nucleotides. If I'm wrong, I will put that in a discussion. But they can be chemically synthesized. So let's talk a little bit about DNA chemistry. So DNA can undergo spontaneous mutagenesis, right? And mutagenesis actually, it's a good and a bad thing at the same time. So for one thing, on one hand, obviously when somebody says that we have a DNA damage, mutagenesis, it's bad because it's carcinogenic, right? Can lead to uncontrolled cell division, can lead to uncontrolled production, production of defective proteins. But it can also be a good thing. Why do you think it could be a good thing? So for example, every day we have 100 cytosines undergoing deamination, this process, which I want to talk in a little more detail about in just in a moment. So we have 100 of these undergoing in every cell per day. So the nature did not design DNA to be completely foolproof, right? So some of this occurs. So why do you think it occurs? What's the advantage of having mutagenesis in our DNA?
Ultimately, it can lead to evolution if that mutation becomes advantageous in some way.
Correct. So if the DNA was totally intact and passed on from one generation to another, then the species would not be able to adapt to the change in environmental conditions, right? Not being able to adapt, and then the species would become extinct if the environmental conditions change too drastically. So DNA mutation is actually a process, as long as it's controlled, as long as it's minimized, and just enough of it is happening to adapt to the environmental changes, then that's actually a good process. So the first one is actually a very interesting one. So here we have cytosine undergoing deamination. So NH2 will be lost and a carbonyl will be gained. Basically, it's a hydrolysis. It's a hydrolysis reaction. Since DNA is swimming in water, there is obviously lots of water molecules which can cause this deamination to occur. And so just think about it. It's possible that the evolution, so the difference between RNA and DNA is uracil versus thymine. And so the evolution selected thymine to be a component of DNA rather than uracil. And probably the main reason for that is, just think about it. Let's say if the DNA did not have this methyl group, right? And had the uracil, just like RNA, then there will be two sources of uracil in the DNA. There will be uracil coming from the DNA synthesis, original, because it's going to undergo base pairing with guanine. No, with adenine, just like thymine, right? And secondly, there will be additional uracil that is produced in the DNA due to this deamination process. And so when the DNA repair enzymes come to basically inspect the DNA and find all these uracil residues, they will not know which uracil should be there because it's part of the genetic code, and which uracil is a mistake. Which uracil is a mistake because it came from cytosine by this spontaneous deamination process that should not have occurred. And that would be the end of the genetic code, right? So we wouldn't. So the DNA repair machinery would not be able to repair the DNA damage. So these mutations will accumulate. These mutations will accumulate day by day, and eventually there'll be so many uracils, so many mistakes in the DNA that will start producing all these defective proteins, multiplying the genetic information from one cell to another, and the organism will not be able to survive. And obviously, the next generation will suffer even more severe consequences. And probably for that reason, the DNA adopted the use of thymine, right? Because now, if there is uracil in the DNA, clearly the only source of uracil is through the deamination of cytosine. And then the DNA repair machinery will come in, take the uracil out, put thymine in, and there we go. And the damage is repaired. Is that clear? It was a long explanation, but it's a good one because we always teach that uracil is part of RNA, thymine is part of DNA, but then the question many of you have asked probably why? Yeah, why do we need to have different? Why is one base different in DNA versus RNA? And that could be a very good reason for that. So there are a couple other deamination processes, right? So methylcytosine, adenine can undergo deamination to hypoxanthine. I showed you this base last time on Monday, which is the precursor for the nucleoside inosine. And guanine can undergo deamination to xanthine. So these are rare nucleic bases and primarily the products of spontaneous deamination. So they can be actually also taken out of DNA and replaced by the original ones if this kind of deamination occurs. All right, so this is deamination. Now there's also depurination. So we talked about this as well. I asked you when we talked about this glycosidic bond cleavage, right? So this bond can be hydrolyzed. So the water can come in and kick out the nitrogenous base. And remember, I asked you which one is more likely to leave, purine versus pyrimidine. And you correctly told me that it was purine because of the number of nitrogens, which can be protonated or can just simply associate with water through hydrogen bonds and become a good leaving group. And so that's why this is primarily depurination. So purines are more likely to leave. And so then we generate this apurinic residue. And there's many more of those occurring in a cell. Many more occurring in a cell. So about 10,000 purines lost per day in a mammalian cell. So the question for you. So how is this damage repaired? So let's say you have a DNA piece in which a guanine was lost. And then the DNA repair enzyme comes in and senses that something is wrong, a base is missing. How does it know that it needs to put guanine back in here? How does it know that it has to be guanine and not something else?
Isn't it just looking at like the other strand of DNA?
Yes.
So for base pairing?
Yeah, yeah, perfect. Very simple. The answer is simple. Right. So it will look at the other strand and find cytosine there and say, oh, it's cytosine, and there must have been guanine here. Right. So the cytosine retains the memory of guanine in the original strand. For that reason, actually, when DNA damage occurs in one strand, the repair is always much easier than if the DNA damage occurs in both strands at the same time, right? So if you cleave the DNA, let's say in both strands at the same time, it's much harder to repair it because you don't know what was there to begin with. And in one strand, it's much easier. Okay, so other mechanisms for mutagenesis are DNA oxidation, right? So here, reactive oxygen species. So we generate a lot of these are generated through part of normal physiology in mitochondria, right? Where oxygen is utilized to burn NADH, for example, nicotine amide adenine dinucleotide. So and that process is not again foolproof. Some of the oxygen will be converted to reactive oxygen species, which can travel to the nucleus and can damage the DNA. And chemical alkylation is another one. So these are some of the molecules that can do chemical alkylation. So the biological methylating agent is this S-adenosylmethionine, abbreviated as SAM. S-adenosylmethionine. And so what happens here, when you have a nucleophile, a biological nucleophile, it can come in, attack the methyl group, and this is a very common SN2 reaction that was studied in OChem1. So now there are a number of other alkylating agents. So these nitrosamines are highly carcinogenic. If you are in a lab and you see a jar or a bottle with nitrosamine, run away because that is carcinogenic agent. The methyl sulfate, nitrogen mustard, also an alkylating agent. The nucleophile can come in and attack, and the chloride will be the leaving group, right? In fact, the nucleophile can attack twice, which can lead to cross-linking. Let's say if this is part of the same molecule, which is DNA, you can cross-link DNA that way, right? Which is bad. Very powerful alkylation process when you cross-link the DNA. But things like nitrogen mustards, for example, they're used to treat cancer. It's interesting. Whatever is carcinogenic is often used to treat cancer, right? So it's actually quite amazing that the same agents which are given to cancer patients also cause cancer, right? They either kill or cause cancer or do both at the same time. That's why actually oftentimes when a patient undergoes chemotherapy, oftentimes a few years later, a patient may have a cancer, a different cancer in a different part of the body, unrelated to the previous cancer. And in many cases that was caused by the chemotherapy. There is chemotherapy-induced leukemia, one of those types of cancers, which is induced by chemotherapy. All right, obviously radiation, UV light. Now for UV light, I want to mention something. Obviously, we know that ultraviolet light causes DNA damage, but the question is how? And one of the main type of DNA damage is the formation of the cyclobutane. Let me draw a structure for you. So this primarily occurs with thymine residues. So they call thymine dimers. And this is one advantage for you to take a class with an organic chemist. So you can actually see the chemical mechanism for this type of DNA damage. So you have a double bond in one thymine. Oh, what did I just do? So we have thymine on the DNA strand. So there's CH3 group here. And there's another double bond from another thymine. Like so. And this can undergo a reaction known as cycloaddition. Guys, do you remember from OGM 2, there was one cycloaddition reaction that we studied in great detail. It was known as a 4+2 cycloaddition. Anybody remembers?
Was that the Diels-Alder reaction?
Diels-Alder reaction, correct. So you have 2 double bonds plus 1 double bond forming a cyclohexene ring, right? Like so. And so arrow goes this way, and this arrow goes this way. And this double bond moves over here. So this is 4+2 reaction. 4+2, known as a Diels-Alder. Diels-Alder. Now, these kind of cycloaddition processes are not limited to 4+2. You see, the reasons why it's 4+2 is because you have 4 atoms on this side and 2 atoms on this side. But thymines undergo 2+2 cycloaddition, and this reaction is actually thermally not allowed. If you take higher organic chemistry classes, this is studied in great detail. 4+2 is thermally allowed, but 2+2 is not thermally allowed, but it's allowed if you irradiate this with light. And if you irradiate this with light, you form a species known as thymine dimers. Thymine dimers. And specifically cyclobutane. There's a methyl group here, methyl group there. Like so. So T dimer. And so not only it links the 2 bases together, right? And it also introduces the conformational change in the DNA. So there's some kind of a kink here because these 2 bases coming together, and that actually makes the DNA change its conformation and kind of move in this direction slightly. So this is the primary reaction responsible for skin cancer. right? So next time you think of putting the sunscreen or going to see a dermatologist, then you can you know actually what process you're looking for. You look what process is being looked at in your skin. Is the process in which the thymine dimers have been formed through the exposure to the sun. All right, so there are a couple other slides that now you already know ATP. We talked about ATP. So for example, these ones. So when you have adenosine triphosphate, you know these are energy rich, right? When you hydrolyze these, so these are phosphodiester, you know these ones, right? So these are energy rich. Energy rich. And so ATP is the main carrier of energy in a cell, right? So when you hydrolyze these, a lot of energy is released. So you know about that. Now, there are a couple other nucleotides playing other roles in a cell. So this is NAD plus or NADH, nicotine amide adenine dinucleotide. There's coenzyme A that is responsible. Basically, it utilizes this sulfur. It's a acetyl group transferrin agent, right? It forms thioesters like this. Flavin adenine dinucleotide. So this participates in the redox processes. But a lot of times you can see these are based on these nucleotide and specifically on adenine. And the reason for that is because adenine is specifically recognized by many enzymes and proteins in the cell. So there is a domain in many enzymes which is designed to recognize adenine, and that's how these things will bind to the enzymes. So the enzymes will bind to the adenine portion and uses this as coenzymes. All right, I think it's good enough. So what we did not have time for is that, as I mentioned to you, Sanger sequencing. So I'll give you pages to read from the text. So 302. There are a lot of kind of additional information in boxes here. Now you don't have to read that, but it's very interesting. So I would suggest you do read that. So pages 302 to 310. So you have that, have a couple problems on sapling. So please read those pages. All right, are there any questions about any of the material we've talked about today? No? All right, then I'll see you on Monday. Right, 10:55, right before 11 o'clock. Let me know if you have any questions. Bye, guys.
Thank you.